Thank you so much. So, we'll our second session. Please welcome our next speaker, Dr. Warren Grayson, who is a professor of development and also a director of the Translation Tissue Engineering Clinic. Please welcome me.
Hi. Thanks for the invitation to speak today. So I'd like to tell you a little bit about some of the work we have going on in our lab. Actually, I wanted to start by maybe introducing the center. I saw a couple other people talk about their centers, so I wanted to start by introducing that. So we've actually just recently rebranded our center to the Translational Therapeutics and Regenerative Engineering. So TTREC instead of TTech, sounds similar. Really to kind of to expand and be more inclusive of all the research we have going on within our center. So, just briefly, this is the center started as a collaboration between the ophthalmology department here and the biomedical engineering department about 15 years ago. We have 10 core faculty members, and bringing collectively roughly about a budget there about 10 million per year. But right now, we've really expanded to have a lot more, not just this our core plus our affiliate faculty that we have. And they come from about 15 different departments throughout the university, in, again, predominantly BME and ophthalmology, but we also have a number of other people from the School of Engineering and the School of Medicine. And the idea, as you may see in the next slide, is that it's really interdisciplinary. Many of us are the core foundational stuff are within cell and tissue engineering, biomaterials, and immunoengineering, but we've also been expanding into sensing, imaging, and AI stuff as well. So some of the faculty that we have here are all computational faculty. And we think about it in the context, so again, those areas that I just mentioned are like those pillars that you would see there, cell and tissue, etc. And we work within all these different areas. Biomanufacturing, disease modeling, looking at healthy aging, and the idea of precision medicine or digital twins. So it's a mouthful to kind of go through all of that. And so when you think about what's the umbrella that everybody falls under, we've called it adaptive therapeutics. And so that name, just to kind of talk about that a little bit more, we look as a convergence, essentially a convergence science. Convergence between tissue and cell on immunoengineering and advanced biomanufacturing with omics, biosensing, imaging, and AI, ML. And we're looking at basically creating these customizable, commercial, and clinically translatable therapies. Before within a number, we essentially collaborate with people throughout the entire School of Medicine. But the clinical areas that we focus on, we have 4 clinical areas that we actually focus on: oncology, ophthalmology, neurosurgery, and plastics and reconstructive surgery. Right. So that's really just to give a flavor of who we are, what we do, and especially in terms of thinking about what kind of collaborations there can be between us at Hopkins and folks within different institutes in Korea. There are a number of people that we can work with. In addition to all the people that I've listed on the slides, we also have clinical affiliates in these different areas as well. So just to talk a little bit more about my own particular research from here. So my lab is really interested in bone regeneration. We've done a lot of work within craniofacial bone regeneration. And I thought I had skipped this slide. So I'm gonna actually just go on to the next slide with this. Here. And we're really gonna focus on the area of biomaterials and delivering these developing biomaterials, bioactive biomaterials that we can use to promote the regeneration of bone in these critical sized injuries, and particularly craniofacial critical sized injuries. Right, so to start with, we think about how to make these biomaterials translatable. And so we, in that sense, we look at things that can be essentially off the shelf in a way, right? So it can be easy to use within the clinic in ways that maybe just harnesses current clinical methods. So in this sense, we've done a few things so far. Just to give a background of and summarize many years of research and many students' research. We've done some work with this study. We're looking at a 3D printable polycaprolactone PCL that's been biofunctionalized with actual bone material, so xenograft bone. And we use this in small, we've done this in a lot of studies in small animal models. What we've gone here is a scale up to a large animal. These are mini pigs, so they're about 200 pounds in size, about the size of a, on the order of the size of a human being. And we create these critical size injuries on top here. So basically create these full thickness injuries within the face of the cheekbones of these pigs and take them out for an entire year. And the first row here basically shows that they're not going to heal unless we treat them. Here we can show the material just by itself promoting this regeneration over time, and you can see this in growth of cells and tissue into this bone here over time. And then if we put the material together with the pig's own cells, so autologous cells from the fat of the pig, we can actually promote even a better regeneration over time. And you can just show this here. So this is one aspect of regeneration that we're looking at within these animals. We've also looked at other materials. So in one case, we looked at how oxygen delivery actually enhances. And this is one method that we have of oxygen delivery. We call them microtanks. I'll show you in another slide, subsequently, another area. But these microtanks are essentially hollow spheres that we could pressurize with oxygen. We 3D print them into the struts of our scaffolds, and so pressurize the scaffolds afterwards. And then once we remove them from the pressurized chamber, oxygen just elutes out. Here we show in mice studies that if we were to put them together with, again, stem cells, just having this oxygen present actually significantly enhanced the amount of bone that forms within these injuries. All right, another area that we think about is another method or material that we worked with is biodegradable metals. In this case here, this is some work we've done with a collaborator in engineering, Tim Weiss, where we've made these magnesium weaves, and we can show by implanting them that we have this ingrowth of vasculature into these weaves as well. And we started modifying all materials to have this PCl together with magnesium to show that you can make these work even better. So we have a number of different compositions and ways that we can modify these materials to make them bioactive. And one of the questions is, how do we think about which areas can we work in? Well, what's the best way to make them bioactive? To study that, we do a lot of interactions, cell biomaterial interactions, and look at those. Those cell biomaterial interactions we use, in particular, is to think about what are the non-osteogenic cells that are contributing towards bone regeneration. And one of the main non-osteogenic cells that contributes towards bone regeneration are blood vessels, right? So we think about ways of looking at and understanding the blood vessel interaction with bone cells. So one approach that we've had for doing this has been using these windows that we could look at regeneration in real time. So think about making this injury on the skull of these mice. If you look at it in cross section, this would be the bone layer here, the bone, and that's been removed, that segment that's been removed. Over that, we put a glass window so we can look in over time, we seal it off. And then we can actually look at this injury as it heals in real time. And this work was done in collaboration with Professor Arvind Pathak in radiology here. So he developed this method. Here you can actually look in at this injury, so you can see the edges of our bone defect. What you're looking at below here is actually the brain and vessels that are through the brain. And over time, you can kind of see how it changes. So this is real-time imaging. Here's where we have some inflammation that occurs. The inflammation resolves pretty early on, around day 4 or so. And then you can start seeing vasculature growing in from the edges, actually up from the surface of the dura mater of the brain as well. And then you start seeing the edges of this defect start to heal with time. So we can take those type of images, even though they look a little bit blurry, you can segment out the vasculature. And with that segmentation, we can look at a number of different things. Here, we're just looking at the saturated oxygen content of the different vessels. We could look at relative blood flow through those vessels, structure of the vasculature, and correlate all of those things with bone formation. So that helps us actually when we start thinking about looking at our different scaffolds. Here we show that we can do this with these scaffolds present here. We just look at PCl only or PCl with a peroxide inside. So this also releases oxygen for a longer period of time. And this is really interesting because we put this in without any cells. And the peroxide itself is actually not osteoinductive. But what you'll see over time is that within 8 weeks, you get significantly more bone formation just by having the presence of oxygen inside of there. Right? We could quantify this. You can see it's about twice the amount of bone forms inside of these injuries, again, just by the presence of oxygen. One one of the questions we've always been asked in terms of that vasculature is, wouldn't oxygen, the presence of oxygen actually inhibit vascular ingrowth? And so what we see is that's not the case, right? So just to look at the early time point and a late time point, it does inhibit it early on. So this is the white bar has the peroxide versus PCl only. But by the time you get to the end stages, you actually get significantly higher oxygen presence inside of it. So a really interesting outcome that you could see with this real-time imaging. And we couple that with other imaging factors to understand whether the cells are playing a role. We've looked at other ways of imaging as well. So this one is light sheet microscopy that's been developed within our lab over the period of several years ago. This is the entire skull of the mouse. We can show is a kind of a way that we can look at the entire vasculature. We could also zoom into different regions of it. And with that, we could also segment out the vessels, the bone cells around them, and look at different interactions between those there. We've taken that and expanded it to look at not just blood vessels, but nerves as well within our imageries. So this is more recent work done by a student who graduated just last week, essentially. And she has been able to show that you can get these, this is the nerves throughout the entire skull. This is in the periosteum, the purple is in the periosteum, the blue is in the dura mater below. And you can see the vessels throughout the entire skull. Sorry. And this is just an optical cross-section. You can see, again, most of the nerves are on the outside, and then they kind of dive in through these regions we call transcortical canals. As we mentioned before, you can do a lot of segmentation out. So these are the teal is the blood vessels, the yellow, the teal is the nerves, the yellow, the blood vessels, yellow and green are blood vessels. We can again look at cell-cell interactions there. So we started to look at how this works in the context of bone healing. Right, so we looked at what I would call subcritical and critical size injuries, show that there's more bone formation, but then we looked at the presence of nerves inside of these injuries, using that same approach that I just described, and found that there's a huge amount of nerve ingrowth in both of them, but the nerves actually persist much longer when they have a critical size injury inside of it. We looked at other cell types present, like osteoblasts, found that the number of osteoblasts osteoprogenitor cells are significantly higher, where there's less bone formation as well, even though there's less of this bone matrix that's being formed in these 4 millimeter versus the 1 millimeter injuries. So it suggests on nerves at high levels, or osteoprogenitors are high levels, bone doesn't form. And so what we wanted to do to understand this better and to really understand the interplay is to look at the signaling between nerves and osteoprogenitors. So to do that, we actually went to transcriptomics. Our challenge in transcriptomics here is that when you start looking at the nerve and the skull, the cell bodies of those nerves are actually relatively far from where the skull is, right? So that's one of the challenges. So we actually have to get two different tissues to do that multi-transcriptomics. So the skull and then the trigeminal ganglia to get the nerves, cell bodies. The other challenge is that the axons from these ganglia, as you can see, there are tons of axons, but they go to all different parts of the skull and midfacial region, right? So if we are trying to get those, if we look at all these cell bodies, we're not actually understanding the cells that are talking to these the bone cells in the skull. So to address that, we've done this retrograde tracing. Phil Wong just introduced this AAV virus that you can use to trace back into these ganglia. With that, we could figure out which cells specifically are talking to the bone cells in our injuries. We've used this approach and shown that we could identify all these different cell types within the parietal bone itself. The parietal bone, all the different cell types there, and the trigeminal ganglia, all the cell types, including the Schwann cells, satellites, glial cells, neurons, etc. Identify which of those cells actually express that tracer from the neurons, which of the cells are expressing the tracer, so we can isolate those out, figure out the differential expression between them. I'm going to skip this for right now, and then look at the crosstalk between those neurons and the parietal cells. So what we're looking at here is this liana signaling that will give us crosstalk, different factors that are being secreted by the nerves and speaking to the osteoprogenitor cells. Several of these factors, about 13 factors, emerged. We started to narrow down from these 13 factors which of these would be important, using an algorithm in terms of which are important in bone, proliferation, differentiation, etc. and then show that FGF1 was one of the main factors that played a role for us. So we did some validation studies with this. This is some in vitro stuff where we took the nerves out, the parietal bones out, did some knockdown studies, so siRNA against FGF, to look at whether or not that actually plays a role in bone healing, right? So what we're interested in, I think the main thing in terms of all of this is that we've developed these really intricate methods, both imaging and transcript, multi-tissue transcriptomics, to look at the crosstalk between these different tissue types present here. And the idea ultimately with this is that you can kind of go back and use that to inform the design of these biomaterials to have even better bone formation. I'm not sure, I might be out of time, but just briefly, we've done this in other tissues as well, right? So our lab works at skeletal muscle regeneration. Using similar approaches here, we could show we can make these engineered fibers. They look very much like muscle. We can do things in bioreactor systems to assess the behavior of these tissues in vitro. We could also implant them in vivo in a volumetric muscle loss injury, show that within these injuries, you get this. Actually, if you look at this histology, it's kind of hard to tell where our injured section is versus the native muscle. Most of this down here is regenerated tissue that you can actually take and look and see the muscle tissues here. Similarly, we can actually start doing these whole mount approaches to look at the entire muscle. Look here, we are interested again in nerve interactions with the muscle. So you can see the entire motor end plate. This is using a transgenic model so that the nerves light up, and you could see the neuromuscular junctions with these. And we've used this now to start looking at what changes occur once we have those injuries in those neuromuscular junctions, right? We can see the difference in nerve patterns and the difference as well in actually the structure of the NMJs, which become fragmented in response to injury. And then when you start treating them, could actually, we can restore these structure and function of these structure, but we don't think function actually of the NMJs. All right, so just to, briefly again, summarize that part where we, and I just wanted to give you a flavor of that we can look at these using imaging, transcriptomics to really start teasing apart novel interactions and using that to inform our biomaterials design. And with that, I just want to thank the lab, the people who've been contributing to these studies, funding, and different collaborators that we have as well. And thank you for your attention.